exciting stuff.
Thank you very much.
Yeah, well,
Now the representative of India.
Good afternoon. Myself, Bhagat Ingole. I am from National Institute of Oceanography, Goa, and I will be presenting the biofuel work done in the Central Indian Ocean under the INDEX program. For the benefit of some of those who are not aware about the INDEX program, I thought I would just tell you a little bit. INDEX program started in 1996-97 initially, and we did the baseline study of the experimental site, and then pre-disturbance and post-disturbance in 97-98, and subsequently we did the disturbance, and then the monitoring was done. And since then we have been visiting the site. I'm not saying at very regular basis because depending upon the cruises, but so far we have done 7 visits to the area since 1997, and this is the site as you can see it. This is Central Indian Basin. This is India and Central Indian Basin, and these are the areas which are overall area which we have surveyed it, and this is the area which is retained area. Red, what you can see is the Indian, and this is the area equivalent area which is the mineable nodal area. Half of the area is relinquished to the Seabed Authority, UL, and this is the area which is with the India, which is called as a first generation mining site, where we are doing more detailed work now. But earlier work, what we had done it was mostly entire Central Indian Basin, and we did experimental study where the disturbance was carried out at 3 km by 300 meter, and 23 stations were sampled, 10 stations on the track before and after. Most of the work what we have done it so far is with the box score as far as the benthic study is concerned. One reason is during 1996-97 we were not having the box score, so we want to have the consistency. We are using the so we are not having multi-core. We do have it now, and so to have the consistency we are using the box score for the sampling. For the meofauna study, as you can see that box score sample we get it like this. It will be divided. We take both macrofauna and meofauna. For meofauna we take it 3 cores of 4.5 centimeter diameter. 2 cores we use it for the preserved with Rose Bengal formalin, and 1 core without the preservation, without the Rose Bengal, so that the taxonomic study can be facilitated. We do have macrofaunal study with the 12.5 centimeter diameter. That sample also will be available for meofauna study, because we usually sample this sea. We use it 37 to 45 micron for meofauna and 300 micron for the macrofauna. In fact, if some of you have seen it, we published a paper in 2009 about the variation between the sieve size for the deep sea in the marine, Journal of Marine Research India, and we have seen it that between the 300 and 500 micron macrofauna, if you can see that more than 40% there will be difference, and therefore final view size, as David Willett mentioned it, that if it is 250 micron for the macrofauna, similarly for meofauna probably we may have to come out with some guidelines, but so far what we have been using is the 45 micron for the meofauna. Okay, these are the representative of meofauna. As you can see that from nematodes we have a different apacticoids, we have ormitids, some of the polypid larval stages, rhinorhynchs we have got it, we have got of course. Now interesting is, I will talk to about it afterward. Okay, when we talk about the composition of megafauna, as you can see that more than 60% is nematodes, and then we will have rest of the various. This is how the megafauna distributed in the entire area. As you can see that there are the patchy distribution. As you can see that there are high densities are there, and these are the points where the box scoring is done. As you can see, covering the entire area. When you talk about the vertical distribution, I was fortunate to have some of the students who work on megafauna. They know it how difficult it is there because it's time consuming. But we did an experiment just for the experiment. It's already published some of the data in deep sea research and marine geotechnology journal. And as you can see that the composition of this was fractured at 0.5 centimeter interval with the core cutter. We had a mechanical core cutter so that we can get to the final section. And as you can see that almost, if you look at it, top 2 centimeter, almost 60% of fauna you can get it in that top 2 centimeter. Of course, we do have some of those nematodes and some of the forms even up to the 40 centimeter, but the time consuming to look for one individual, you have to see what is at your disposal. So depending upon that, you can. This is how what we did it. For example, pre-disturbance and post-disturbance. This was the inside the track. As you can see that this was the disturbance track, and this is 3 kilometer by 300 meter width. This was there, and these are the stations which are all those box scoring which are located in the entire area. So we have both side of the track and on the track the sampling was done. So pre-disturbance and this post-disturbance. There was 45% loss immediately after the disturbance, which has not been recovered even till today. As we did a cruise in this November in the area, samples are not yet analyzed, so I don't have data to share it. But what we know it from over a period of time, the recovery is very slow. If at all it is going to take place for some of the groups. As far as the nematodes and hyperacticoids, we had taxonomists with us, so we could do it some work up to the genus level, some of those things. And here we have got it. Nematodes, for example, these are the nematodes families, some of them, some of the genus. What we have got it here. These are the 4 or 5 dominant families are there. There are some species yet to be described. Probably could be the new species. Also a new record to the area. I'm not saying that they are new species because we need to study it in detail. Hyperacticoid, as you can see, that these are those stations, as if you can place it in the area, and then how the diversity of the composition is the different families of the species distribution. If you see north to south of the area, if you go from north, like 9 degrees south to the 17 degrees south latitude, and this is how it is. There are the patches, again the high diversity of some of the areas you do have it as far as nematode is concerned. And also herpetocoid, a similar pattern is there, but of course in a different type of pattern. We do get high diversity at some of the locations. That's what the patchy distribution. What is most important, which we have been talking since from yesterday, there are some of the organisms only, some of the species, one only organisms we could get. So doing it molecular study or those things, we were not thinking at that time, because it is important from taxonomical point of view. When you have got 1 or 2 specimens, how to distinguish with whether you want to go to molecular or you want to go for the morphology or preserve the halotype specimen. What you want to do, you have to decide about that. Okay. This is about the tanids. There is one more component which we have added recently, is about the nodule associated fauna directly. Nodule as a substratum, as you can see there. Of course, it's not a megafauna, I just wanted to show it mega and macrofauna, how they are associated with. And these were some of the groups which were in nodule and in the sediment, as you can see that. So this is in the nodule and this is in the sediment, but there's just preliminary study. We did it along with the geologists, so what they help us to classify or identify type of nodules. So they gave us the classification, 5 type of nodule, but we get it in the central Indian basin basically, and then depending upon the different type of nodules, what we did it, from each box score we used to collect, before, when we open the box score, before disturbance, minimum 10 nodules of the assorted nodules, we'll take it separately, and those we'll be using for neofauna. Immediately that will be preserved in West Bengal formalin, and then very carefully it will be washed with fine brush to remove all the sediment, whatever is there, and fauna associated with that. That will be preserved, and then carefully in the laboratory it will be analyzed. Nodule as it is will be measured for the dimension after identifying type of nodule to the surface area, basically to find out how much is the surface area, and is there any relationship between that. So here is some, for example, from the mining point of view, they, if I'm not wrong, this is what I understood, is that they say that there are nodal poor area and nodal rich area. If you see nodal rich area, something like 10 kg per meter square could be the the area which the area can be used for the mining, and less than 2 it can be called as a poor area. That's what my understanding is there. I may be wrong. So this is how what we have done it. For example, you have got the nodules from the nodal rich area and nodal from the nodal poor area, and here is the group density, particular diversity, group number of meiofauna taxa would be get it associated with it. This is how it is there. As you can see, it is more associated with in the nodal, and here is again what you have got the nodal density, different density. Here is the density of fauna associated with the nodal, and as you can see that here is the nodal surface area, and here is the abundance, and as you can see that it certainly shows the positive relationship with the surface area. That's what the preliminary result what I can say. In summary, nematodes abundance recorded in Central Indian Basin certainly is lower than reported elsewhere. Nodule and sediment associated morphotype, we have got it almost 284 different nematodes belonging to 30 families and around 113 genera. There are 3 families which are most dominant, like as you can see that Comedoide, and then this one which is most abundant among the different families. The diversity varied significantly from different stations, as I have shown it how it is distributed from north to south. The diversity varies it. There are the patchy distribution, particularly some areas we have got high diversity. When we look at it carefully about the population structure of nematodes, there are more than 65% female dominated in the samples. Could be that could have been the I don't know whether it is a breeding season or something like that, particularly when the sampling was done. But this is what was the observation. And finally, the most of the data is already shared it since 1997, shared with the OBs, also published around 30 papers, macro and meiofauna together, around published in DSR +1, MBR, JMBA, Zootaxa, Zookeys, and is marine geotechnology and geochemistry journal, environmental monitoring and assessment, and also has been used by various people because it's available in the OBs, people for global synthesis. I think so. This is what I have to say as far as the meiofauna is concerned. And after I had collected this specimen almost 20 years back, I can say that it is lying on the slide. When I visited Pedro in 2005, Pedro's laboratory, I had carried it with me, and then I was in Oldenburg for some time. They said that probably it is the Lolicifera, but I was lazy a little bit. I didn't bother much about it. After Professor lecture yesterday, I really got inspired. I might be sharing this sample with you. Thank you very much for that.
Questions?
I finally wanted to thank my PhD students who have been my workforce for the data collection, samples, analysis, then International Seabed Authority to make it available, at least this forum to me, organizers and others, and of course the Ministry of Earth Sciences, Government of India, who is the funding agency for this work.
Could you go back to the slide where you showed the sampling of the area? Please.
Sure.
Just that, yeah, that would, the one with all the, yeah, maybe that.
Yeah.
This is a very big area, isn't it?
Yes. I think so I can. Yeah. This is, if I'm not wrong, 150,000 square kilometer. This is how it is there.
So, right. Okay.
That entire area. Yeah.
So how would you? Let's imagine if you wanted to harvest the nodules from there. What would you do?
I don't know what to tell about the geology or mining. I'm a biologist, so I will not be good on my part to comment on the mining.
Right. Okay.
Yeah.
Well.
Because I'm not aware about it. To be safer, that's what I meant. Yeah, Pedro, I know.
Thank you for an excellent talk. Could we get the last slide again?
Yeah, sure. That is what I also want. I also want that you should have a look
Yeah,
at.
sure. This is extremely interesting. It's not a dossier for us, so I think it's a cnidaria.
That is why I didn't put the name after talking to you.
Yeah.
I put it. I had put a question mark.
No, it's fine.
Yeah.
But I think it's cnidaria, and the reason I think that is you can see the cnidoblast. But of course, I have to examine it better. But this is very interesting. If you found that on the surface of the
So going back, I will go back to my collection, see the slides, and if it is available, I'll be happy to send it.
Thank you.
Hope it is available.
Thank you.
But I have also seen in the other presentations, I think we need really to make some standardization of the taxonomy. So, we need a workshop to talk just about the copepods, maybe another workshop just about the nematodes, to use the same names for the same animals.
Slovakia.
It's not a joke. It's extremely important. Even if you consider that we're moving forward to barcoding, we need also to give the right names to the barcodes. Otherwise, it will be a mess because we will not be harmonizing. What? And this is what we need workshops, and this is why we need this kind of workshop where we really come different contractors together with the material and try to make harmonizing the taxonomy.
In fact, I just wanted to make a comment. Most of these studies were done jointly with the geologists. It was always very difficult for me. It's a practical problem I'm talking about. Maybe with me, I don't know about others. Always very difficult to get the replicate sample for me. If I wanted 2 cores, they will be, they said, no, why you want one is enough for you, why you want. So, in fact, I was fighting to get full box core. And the working at 6 kilometer and get a full box core, it is economically also and time. Both are the things that are there. So, it was very difficult initially to get a full box core, particularly for macrofarm. But now, I can say thanks to the Seabed authority, they have made it some points at some level, and last cruise we could get some full box core. Earlier also I got a few of them. Also, multi-core will be using it. We have used it in some of those things. Maybe if I have good data, in the future we'll be able to compare it box core and multi-core.
Can you go back to the map where you showed the densities in different colors?
Yeah.
I just wondered, that one, how you made the extrapolation. Was it based on environmental conditions?
Yes, yeah. Sediment type, basically. It was
Based on sediment type, you
yeah.
extrapolated?
Yes,
Yeah.
yeah.
And I mean, such an approach is useful, but we need to be sure that the full scope of environmental variables that possibly drive the density.
What actually we have done is some of data is already shared with the Seabed authority. We have geochemistry data, we have microbiology data, we have biology data. All the data with the same scale we have used this map. And so that we have got it across the biology, geology, geochemistry and microbiology data, so that we can do the comparison, which parameters. So that's what the purpose of doing this.
Yeah, I think the remark might be being made is that by using a regular grid for setting where the samples are taken, however many variables you take, may miss the heterogeneity of the environment, because you're not sampling depending on where the greatest variation might be. So many of the other studies use, say, bathymetry, spot bathymetry of the seabed to see where the greatest variation in the environment is, and then focusing the sampling on those areas, and where there's more homogeneity, then you can have fewer samples. So the question I think I'm hearing is that grid way of approaching things really appropriate to the environment that you're working with on the sea floor, which is really a series of ridges and troughs.
If I can just say something about, for example, in this case, we visited the site only which is disturbed site, and that's what we call as an experimental site and reference site regularly. But rest of the area where we have done the sampling, it is definitely based on the bathymetry, and we will see that where there is a variability, among that we will place the box score and we will connect this. So environmental variability is very much taken into consideration while selecting the location.
And can I make just one other thing? It's great to know that what the ISA has told your government has allowed you to have some more sampling.
I ought to acknowledge that. That's what I said. Yeah.
But it does.
But they were not listening to me when I was in the discussion. They said, why do we need it? Because it is very expensive to give 10 box score means you have practically 4 to 5 days of working time. So 5 days of working time in the deep sea is very expensive. Only for the macrofauna if you want it, if you see it from that perspective. But now they have agreed for it.
Yeah. But I still think this workshop has to think about the conclusion that came from the macrofaunal workshop was, and this is to convince those that are in policy, in governments, which don't really understand about the variability of the deep sea floor and the biology and how you measure that and how much work goes into it, that the recommendation from macrofauna was that there should be biology-specific sampling cruises, not just the occasional sampling. And
Thank you for that.
You may be wanting to make that point yourself and discuss it within this workshop. That may be a recommendation that you still want to be making in relation to Myofauna.
I fully agree with you, and I thought that type of recommendation should be given by ISI to the contractor, so that if it is required, they can be specific biology, at least oriented course, or basic biology oriented course.
Well, just to follow on from what David said, one of the other recommendations that came out of the macrofauna workshop was that the experts highly recommended that there is no subcoring within box cores because the issue is that there's not enough fauna anyway, and the moment you start to subcore, you're actually destroying anything you're going to get. So that was a very strong recommendation that came out of the workshop as well. So it's something else to take back to you.
I do agree with that, but then when you have to treat the full box score, we have to subsample it. You can somehow will have to divide it, and you cannot full box score. You cannot even see it. You cannot preserve it. It has to be taken at least in part. So
yes,
if you
but
can,
we're not suggesting that you. Not for the
you don't consider as a replicate. I understand it. Yeah.
What tended to happen in some cruises was that everybody went into the box core, took their bit, and disappeared,
Yes.
and then all that was left was, oh, you can have it for the macrofauna. What we
It
were
happened with
suggesting
me. It
is
happened with me. I agree with you.
you can split it up to
Yes.
wash it, but
In fact, I have used that type of sample. We do have it. I didn't comment about the barcoding, but we have used that sample for the quantitative analysis, and maybe if it is preserved in the code, we will be using it for the barcoding.
Yeah.
This graph you show there is very different from all what we've seen the last 2 days. And what is really different is the helicarry. It's the first time we see.
It is not helicarry, it is orbited, basically.
Yeah, I know.
And
Most of them would be
yes,
helicarry.
yeah.
But take a look, 18 to 20 centimeters down in the sediment.
Yes,
It's very deep.
yes, yes.
So perhaps some of the other are lost. It's very, very deep in the sediment.
In fact, some of those, in one or two tube, I have got the live polychip tubes down up to the 20-25 centimeter
Okay.
tube. So I don't know whether that facilitated the burrowing or the migration.
It could be
that type of thing.
burrowing.
But that's, yeah. Because we got the live burrow and the polychip was available at the down. But that we had one or two only tube we could
But
get.
it's really unique.
Yes.
There are so many. So
And I would like to share it not from this. In one of the area we did it in Andaman Sea, around 2,500 meter. We got halacarids were the dominant both the nematodes and hermaphrodites at one or two stations. Only one of them, maybe that type of patch or something was there, but rarely that type of things was observed.
Sure, sure.
I had put up that type of, I think, poster in one of the deep sea biology symposium where David was organizing in Southampton. That poster I had put it.
Thank you.
Thank you very much.